Hello, in this video we discuss three questions. First one is significance of HMP shunt pathway. When you write about the significance of HMP shunt pathway, you have to write about the physiological significance and the clinical importance. HMP shunt pathway, it has two phases. One is the oxidative phase and the other is the non-oxidative phase. The main purpose of the HMP shunt pathway is to produce ribose phosphate and to generate NADPH. The oxidative phase of the pathway produces NADPH, and the oxidative phase occurs in liver, adipose tissue, adrenal cortex, mammary gland, testis, ovary, red blood cells and lens of the eye. The non-oxidative phase, it is present in all tissues, so synthesis of ribose is possible in all tissues of the body. The NADPH, which is produced during the oxidative phase, it is used for the reductive biosynthesis of fatty acid, cholesterol and steroid hormone. So, the major metabolic role of this pathway is to provide cytoplasmic NADPH for reductive biosynthesis of fatty acid, cholesterol and steroid hormones. Then NADPH, it is used for free radical scavenging. That is, this free radical, they are continuously produced in the cell. They will destroy DNA, protein, fatty acid and biomolecules, and in turn the cell will be destroyed. So, this free radical, they are inactivated by enzyme systems like superoxide dismutase, peroxidase and glutathione reductase. To regenerate reduced glutathione reductase, NADPH is required. Then NADPH is required to maintain the RBC membrane integrity. So, NADPH, it is required by RBC to keep the glutathione in reduced state, and this reduced glutathione, it will detoxify peroxide and free radicals which is formed within the RBC. So, NADPH, glutathione and glutathione reductase together, they will preserve the integrity of RBC membrane. Then NADPH, it is used for the prevention of methemoglobinemia. That is, NADPH is required to keep ion of the hemoglobin in reduced ferrous state. So, that will prevent the accumulation of methemoglobin. Methemoglobin, it cannot carry oxygen. Then NADPH is used for the detoxification of drugs. Most of the drugs and other foreign substances, they are detoxified by liver microsomal P450 enzyme with the help of NADPH. Maximum concentration of NADPH is seen in the lens of eye, and NADPH, it is required for preserving the transparency of lens. Then NADPH is also required for the macrophage bactericidal activity. That is, NADPH, it is required for the production of reactive oxygen species by macrophages to kill bacteria. Then the non-oxidative phase of HMP shunt pathway, it is for the production of ribose, and it occurs in all tissue. So, this ribose and deoxyribose are used for DNA and RNA synthesis. Then the clinical importance of the pathway are glucose 6-phosphate dehydrogenase deficiency, drug-induced hemolytic anemia, methemoglobinemia and thiamine deficiency, which leads to the reduced activity of transketolase enzyme. The enzyme glucose 6-phosphate dehydrogenase, it may be deficient in some person. So, the defect is transmitted as X-linked recessive trait. This deficiency, it will lead to drug-induced hemolytic anemia, and the deficiency is manifested only when exposed to certain drugs or toxin. For example, intake of antimalarial drugs like primuquine. So, this primuquine, it will stimulate peroxide formation inside the RBC. In glucose 6-phosphate dehydrogenase deficient cell, the level of NADPH is low. So, the further production of peroxide, it will lead to the lysis of cell. And similarly, intake of toxic glycoside, which is present in the fava beans, also have the same effect. Then sulfur drugs, they will also precipitate the hemolysis. So, this will lead to jaundice and severe anemia. Next one is methemoglobinemia. So, this glucose 6-phosphate dehydrogenase deficient person, they will show increased methemoglobin in circulation. That is, NADPH, it is required to keep the ion of hemoglobin in the ferrous state. Since NADPH is deficient, there will be increased methemoglobin in circulation. Then thymine deficiency, that is the transketolase enzyme. Transketolase enzyme, it is dependent on thymine pyrophosphate. So this transketolase reaction, it is measured in RBC as an index of the thymine status of the individual. And the occurrence and manifestation of Wernicke-Korsakoff syndrome, which is seen in alcoholics and those with thymine deficiency, is due to the defect in enzyme transketolase. Second question is digestion and absorption of carbohydrate. The whole process of digestion and absorption of carbohydrate, it occurs in a well-defined sequence. So in diet, the carbohydrate, they are present as complex polysaccharide like starch and glycogen, and some amount of disaccharide like sucrose and lactose is also present. So this polysaccharide and disaccharide, they are hydrolyzed into monosaccharide. So the process of digestion of carbohydrate, it begins from the mouth. In mouth, the saliva contains an enzyme salivary alpha amylase. So this salivary alpha amylase, it is an endoglycosidase which will hydrolyze the alpha 1,4 glycosidic linkages. So the polysaccharide, they are converted into dextrins by the enzyme salivary alpha amylase. The digestion of carbohydrate in the mouth is very limited because the time available for digestion in the mouth is limited. That is, the food remains in the mouth for a shorter period. Then it comes to the stomach. So the carbohydrate digestion in stomach is temporarily stopped, that is because of the high acidity. So this high acidity, it will inactivate the enzyme salivary alpha amylase. Then the further digestion of carbohydrate, it occurs in the small intestine by the enzymes, by the pancreatic enzymes. So this pancreatic alpha amylase, in the pancreatic juice, there is an enzyme pancreatic alpha amylase. So this pancreatic alpha amylase, it will hydrolyze alpha 1,4 glycosidic linkages to produce smaller subunit like maltoses, isomaltose and dextrins. So dextrins, they are converted into maltose by the enzyme pancreatic alpha amylase. This occurs in the small intestine. Now the cells of brush border of intestine, it contains enzymes like sucrase, maltase and lactase. So this maltase enzyme, it will convert maltose into 2 molecules of glucose, and sucrase enzyme, it will convert sucrose into glucose and fructose. Then lactase enzyme, it will convert lactose into glucose and galactose. So this is the digestion of carbohydrate. So polysaccharide in the mouth, first they are acted upon by the enzyme salivary alpha amylase which convert it into dextrin. Stomach, there is no digestion because of the high acidity. Only some amount of sucrose is converted into, by the action of HCl, some amount of sucrose, it is converted into glucose and fructose. Then pancreatic alpha amylase in the small intestine converts dextrins into maltose. Then the brush border of the intestinal cells contain different enzymes like maltase, sucrase and lactase. So this will convert maltose to glucose, sucrose to fructose and lactose to galactose and glucose. Coming to the absorption of carbohydrate, only monosaccharides are absorbed by intestine, and the absorption rate, it is maximum for galactose, moderate for glucose and minimum for fructose. Glucose has specific transporters which are transmembrane proteins. Absorption of glucose, it involves different mechanism. So the absorption in the intestinal epithelial cell, it is by carrier mediated transport mechanism, and this is an energy dependent process. So absorption from intestinal lumen into the intestinal cell is by co-transport mechanism. That is, a membrane bound carrier protein, it will carry glucose along with sodium. The co-transport is with the help of carrier protein sodium dependent glucose transporter 1. So this sodium dependent glucose transporter, it has 2 binding sites, one for the sodium and the other for glucose. So binding of sodium, it will result in conformational change in the carrier protein. So, this change in the conformation that will enable the binding of glucose. Then sodium diffuses into the cell, dragging glucose along with it. So, this is an energy requiring process, and the energy is indirectly provided by ATP. Transport of glucose into the bloodstream and to the peripheral tissue is conducted by some carriers called glucose transporters, and they are sodium independent transporters. So, the same intestinal epithelial cell, they have a different transport mechanism on the membrane facing to the capillaries, that is on the basal surface. So, the intestinal cells, they will release glucose into the bloodstream by the carrier mechanism called glucose transporter type 2. So, this transporter, it is not dependent on sodium, and this is a uniport facilitated diffusion system. GLUT2, it is involved in the absorption of glucose from bloodstream to the cell, and this GLUT2, it is present on the intestinal epithelial cells, liver cells, beta cells of the pancreas and kidney. So, this GLUT2, it will enable the pancreas to monitor the glucose level and adjust the rate of insulin secretion. Then glucose transporter 4. This glucose transporter 4, it is the major glucose transporter which is seen in skeletal muscles and adipose tissue, and this is under the control of insulin, and the other glucose transporters, they are not under the control of insulin. Insulin, it will induce intracellular glucose transporter 4 molecule to move to the cell membrane and increase the glucose uptake. The other glucose transporters are GLUT1, which is present on the RBC, brain and kidney. So, it is involved in the glucose uptake in most of the cell. GLUT2, as we discussed, it is present on the basal surface of the intestinal cells, liver, beta cells of pancreas. So, it is involved in the glucose uptake in liver, and it is the glucose sensor in beta cell. Then GLUT3, it is present in neurons and brain, and it is involved in the uptake of glucose in brain cells. GLUT4, it is seen in skeletal muscle, heart muscle and adipose tissue. So, it is under the control of insulin, that is, it is involved with the insulin mediated glucose uptake. GLUT5, it is seen in small intestine, testis, sperm and kidney, and GLUT5, it is the fructose transporter, and it has poor ability to transport glucose. Then sodium dependent glucose transporters, it is seen in intestine and kidney. Third question is lactose intolerance. The enzyme lactase, it will hydrolyze lactose to glucose and galactose, and this enzyme, it is present in the brush border of enterocyte. The deficiency of lactase, it will lead to lactose intolerance. So, the enzyme deficient in lactose intolerance is lactase enzyme. When this enzyme is deficient, lactose get accumulated in the gut, that will lead to irritant diarrhea and flatulence. So, this condition should be recognized and treated in newborns by giving lactose free formula diet instead of milk, and there may be congenital or acquired causes for this. Acquired lactose intolerance can be due to the sudden change into a milk based diet, and this enzyme lactase, it is an inducible enzyme. So, milk could be withdrawn temporarily and the diarrhea will be limited. The symptoms of lactose intolerance include diarrhea, flatulence, abdominal cramps and distension, and lactose will appear in urine called as the lactoseuria, and the treatment is to give lactose free diet.